welcome everyone. My name is Chris Edgar. I'm the Senior Vice President of Clinical Science with Cogstate, and today we're going to be looking at the value of assessing cognitive safety and tolerability throughout the drug development process. The term cognition covers multiple processes, and these may include perceptual, attentional, information processing, memory, decision-making, and motor control. It's important to consider multiple assessments which cover domains of relevance when we think about concepts of interest for measurement of cognition. There are several models of human cognition in existence and that give us insight into which are the major domains of human cognition and those elements of cognition that may be vulnerable to change. However, as drug developers, we need to balance comprehensive assessment against trial burden when selecting test batteries. In respect of determinations regarding the construct of cognitive test batteries, FDA guidance can also be used to provide some useful insights. When we think about the assessment of safety and tolerability in clinical drug development, it's really critical to explore that carefully in terms of the need for such assessments and how they're conducted. There are many CNS and non-CNS compounds that have the potential to negatively impact cognitive ability, and particular drug classes may be associated with greater risks, and these are things like blockade of muscarinic or histaminergic or beta-adrenergic receptors. Drugs can have multiple pharmacological effects, some of which are desirable, what we might term on target, but many of which may be undesirable or off-target effects, and these can include cognitive dysfunction. Also, given the increasing awareness amongst regulators and the general public, assessments of cognition are widely employed then to examine the potential for short and long-term cognitive risks. An assessment of cognitive function then becomes an integral part of the decision-making process during clinical drug development. We see then that data on the potential adverse cognitive effects can be informative in several ways. They can actually help us in determining dose-response relationships and selecting the correct and safe doses for later development, including the potential need for dose titration. They can act as differentiators from competitor drugs in relation to cognitive safety and tolerability. We can help to detect off-target pharmacological effects. And also we can look in general at the risk-benefit ratio in relation to the target indication itself. In addition, it might also be worthwhile to continually assess cognitive safety and tolerability following approval and marketing via the longer-term monitoring of cognitive function as part of routine pharmacovigilance. There are multiple case studies in the literature around specific classes of drug that have shown a potential to impair cognition, and for which extensive evaluation across different populations has been undertaken in order to attempt to identify vulnerable groups and demonstrate the absence of adverse effects with perhaps different formulations or more specific mechanism of action. Here we can see multiple examples looking at the use of anti-muscarinics in the treatment of overactive bladder, where it's been identified that some CNS active drugs may cause cognitive impairment, and where the risk of cognitive impairment may be far greater in the elderly, particularly the elderly with existing risk factors for cognitive impairment around the use of anti-muscarinics or related to diseases like Alzheimer's. But two, that there can be peripherally acting drugs or different formulations that may mitigate many of these concerns around cognitive impairment. If we shift then to thinking about the regulatory guidance landscape, there are many contexts in which objective performance-based assessments of cognition could add information to clinical drug development, and this information be relevant in terms of its generation across multiple phases of development. In general, the inclusion of targeted objective cognitive assessments into clinical development programs will be prompted by different kinds of information that emerge at different stages. Early on, we may have insights from mechanism of action and preclinical data that could suggest the potential for adverse cognitive effects early on, and the value of inclusion of objective tests in our earliest first-in-human single or multiple ascending dose studies. Additionally, we may see spontaneous adverse events or clinical observations during early development, prompting more targeted investigations in later studies. Alternatively, we might see that later in clinical development that there are clinical populations, such as pediatric or elderly populations, where novel safety signals may emerge. There may be the potential for certain drug-drug interactions or comparisons with other treatments that all come together to prompt, again, more detailed evaluation of cognitive function. The FDA have long recognized the potential of drugs to cause cognitive impairment and for effects on cognition to be important as a guide to general safety and tolerability. The guidance on hypnotic drugs from 1977 discussed in several places evaluation of effects, including residual effects on alertness, mood, judgment, coordination, reaction time, and memory. And we know that there are other important guidances from FDA looking at, in a particular, abuse liability and also ability to operate a motor vehicle, which focus on the objective assessment of cognition. Then too, we see that there are guidances which deal with longer term safety from both EMA and FDA, and which may be focused on particular areas of development, such as looking at efficacy and safety in pediatric medicine development, or specific to the development of drugs for migraine. So next we move on to look at how data from objective performance-based assessments might be integrated with different sources of information regarding cognition. In general, then, we have three primary sources of information regarding the potential negative effects on cognitive performance of different drugs. These are spontaneous adverse events, self-reported assessments or patient-reported outcomes, and two, performance-based outcome measures, so cognitive tests. Adverse events are key safety outcomes, and the use of preferred term buckets can provide important clinical insights into potential CNS effects. Importantly, though, adverse events may not provide much granularity around the timing and magnitude of effects or targeted information about the nature of such effects. And this can be particularly important for cognitive impairment, which FDA recognizes difficult to report and observe. Self-report tools can provide useful additional insights that structure findings around concepts like mood and alertness or sedation, as well as provide data with increased granularity around timing and magnitude that can be subjected to more detailed statistical analyses to draw out, for example, PK-PD relationships. FDA recognized too that in assessing cognition, self-report may be inadequate. Classically, alcohol is known to impair driving ability, but may not impair perceived ability. Performance-based outcome measures have the same advantages as self-report, with the addition of being free from biases and also measuring additional cognitive concepts that cannot be easily assessed via self-report. Many test paradigms exist with which to assess recommended domains with respect to ability to operate a motor vehicle, for example. Importantly, we'll discuss shortly objective performance-based assessments, i.e. cognitive tests, as opposed to observation, spontaneous adverse events, or self-reports. Here we can see an example of a computerized performance-based cognitive test. This test is called the identification test and is a test of choice reaction time and measures attention. A test like this is brief and sensitive, but also highly reliable and stable with repeated assessment, and can be particularly useful for looking at the potential to adversely impact attentional performance over a period of time with a drug, such that we can understand the onset of drug effect, its potential peak, and also the time of offset, and begin to generate a clear understanding of how domains of cognition may be impacted. And importantly, domains of cognition which have relevance for important aspects of function like ability to operate a motor vehicle. Here we can look now at an example of the use of performance-based cognitive assessments to detect effects of a drug which may not be measurable via self-report or spontaneous adverse events. In this particular study, a battery of performance-based cognitive tests was administered to healthy male volunteers in a simulated phase 1 study using midazolam to induce sedation-related adverse events, but also cognitive dysfunction. For each of the subjects, the magnitude of cognitive change between pre-dose and 1 hour post-dose was calculated, and group and individual level cognitive outcomes were compared between subjects who either did or did not report cognition-related adverse events, such as sedation, following the administration of either 1.75 or 5.25 milligrams of midazolam. Effects of midazolam were apparent across several outcome measures, including psychomotor speed, attention, executive function, and learning and memory, but were also evident in self-reported adverse events. Although the size of the effect on performance-based assessments was smaller in those not reporting cognition-related adverse events, in many cases, the effects were still large, with effect sizes of 1 or greater, thus indicating that there are important effects of midazolam on cognition, which may not be apparent to the individuals receiving the drug themselves. At both doses, there were subjects who did not report cognition-related AEs, but who were still impaired in their performance on the objective tests, defined as an effect size change of 1.5 standard deviations or greater. Thus, it can be seen here that reliance on spontaneous adverse events has the potential to miss relevant information regarding cognition. We'll look now at key considerations for selecting and optimizing computerized cognitive assessments. Here we can see an example of a hidden pathway maze learning test, and this is a measure of decision-making or executive function. One of the important elements of successful cognitive tests for the assessment of safety and tolerability is that the tests themselves, and consequently the test batteries, must be brief, they must be multiply repeatable, and they must be stable over time in the absence of treatment. And this is important in order to be able to detect the onset of drug effect, the offset of drug effect, and also the potential peak effect over the course of either hours or days, such that we can look at the course of a drug effect over relatively short periods of time and in healthy individuals. And also that we have tests which are bidirectionally sensitive, such that we can see either improvement or worsening performance, again, in individuals who may be healthy. What we can see here is an example of two different drugs, one intranasal esketamine, the other lorazepam. For both drugs, we can importantly see that in the placebo arm, where there's no treatment, performance remains stable over time with repeat testing, and there's little change happening in the placebo arm. However, for both of the active drug treatments, a clear impairment is evident in terms of an increase in errors in performance on the test. And we can see that increase in errors peaks at a given time point, but also recovers over time. And we can see here the utility then of the cognitive test in being able to detect that effect, its time of peak occurrence, but also the time to resolution. And these kinds of information then can be important components in our evaluation of safety and tolerability, and at what point individuals have recovered to a point at which they can continue to undertake their normal activities of daily living. Another critical component for our understanding and evaluation of different kinds of cognitive tests is the extent to which we can look at the results from the test and place them into context. Here, again, effect size can be an important metric for us. We can use effect size in order to compare a drug effect that we see to common effects that emerge with different kinds of drug treatment or other kinds of benchmark that might be valuable to us. These include things like blood alcohol concentration. We know blood alcohol concentrations are reliably associated with motor vehicle accident risk. And in addition, other kinds of sedative hypnotic drug or the effect of sleep deprivation have relevant effects that can be used as useful benchmarks for interpreting our data. And this kind of context setting is particularly important when we want to understand the relevance of a treatment outcome. We'll now move on to look at lessons learned from global cognitive safety studies. To evaluate cognitive safety and tolerability longer term, acceptability for patients can be critical, especially for patient populations who may find such assessments more challenging due to reasons of cognitive and motor impairment or fatigue. In patients with multiple sclerosis, the Cogstate computerized tests have been evaluated for acceptability versus traditional assessments, including the paced serial addition test, or the PASAT. In the study illustrated here, cognitive test data were gathered for 37 participants with relapsing remitting MS over a period of 12 months, and additional acceptability data were gathered. Whilst both the Cogstate computerized tests and the paced auditory serial addition test discriminated between MS and healthy controls at baseline, the computerized tests were more acceptable and caused less anxiety. The duration of the computerized test was rated as not too long to just right by 86% of participants, compared to 50% for the PASAT, while 75% of participants were happy to repeat the computerized tests, compared to 52% for the PASAT. Performing the PASAT caused anxiety in 69% of patients, compared to 41% for computerized, and test enjoyment was rated as neutral to highly enjoyable by 83% for the computerized tests, compared to 61% for the PASAT. Such data then are useful in supporting the feasibility of assessment over longer durations. It has been established that cognitive dysfunction may affect up to 65% of multiple sclerosis patients and also worsens over time. In this example, we see data from a single-center 24-month observational study in 63 patients treated with natalizumab, who were assessed prior to monthly infusions using a Cogstate computerized battery and also the symbol-digit modalities test. As can be seen here, data were collected on a monthly basis for a period of 2 years. No patient showed evidence of sustained cognitive deterioration over the 24-month period. The data was split into groups based on time on natalizumab, under 2 years or greater than 2 years. Irrespective of time on natalizumab, significant improvements were observed in executive function, verbal memory, and working memory. These results suggest that natalizumab may preserve cognition with continuous therapy. This also illustrates the feasibility of long-term safety monitoring using computerized cognitive tests and in this particular patient population. In summary then, the assessment of cognition can provide valuable information across multiple stages of drug development and for a range of planned and targeted trial designs. A stepwise consideration of cognitive safety and tolerability may be applied, which can use emerging evidence related to the hypothesized mechanism of action of a drug, adverse events that may emerge through the different trials that are conducted, and also subjective effects on patients in certain populations. Cognitive decline measured using objective cognitive test performance is an important indicator of potential adverse effects, and safety decisions can be based on the extent to which cognition declines in individual subjects. Information about a decline in cognition should guide decisions about drug safety in the context of other available information, like adverse events and self-report. Integrated digital solutions can dramatically streamline this assessment, reducing site and patient burden, as well as increasing sensitivity and precision of measurement. Thank you for listening to this brief overview of considerations for the assessment of cognitive safety and tolerability. We hope the presentation was informative and look forward to your questions.